Yeah, hi students. Welcome back to my video class, online video class. And our topic is clinical study design. The clinical study design, it is my second video. So you go and see my first video. Okay, clinical study design. It will be there. Okay, in my playlists. Okay, it will be there. This is my second video. In my first video of clinical study design, I have explained some slides. Okay, see here. This is a unit 1 structure. These are some my previous slides. You try to recall all this. Yeah. So exactly here I stopped. Okay. Case control study. See here, case control. Case control. Study based. See this structure. Why this is called? It is just like a cohort study. This case control study. Cohort study already I have told in my previous video that smokers are there, non-smokers are there. Okay. See here in study base cases, controls. Here cases means smokers. Controls means non-smokers. They are not having a smoking habit. No. Say no to smoking. Controls. They are under control. Okay. Whereas a cases means case gone. That's it. Okay. They are the smokers. Okay. Non-smoker. And here I have told those who exposed and unexposed to lung cancer. Okay. Here 40% they exposed to lung cancer, they got disease, and here 10 members not got a lung cancer because chances are very less, no? And here exposed, if only 15 members got lung cancer, even though they are not smoking because of hereditary or by birth some genetical problem or from their parents or from the industrial pollution and all, they got a lung cancer, okay, and unexposed. And here odds ratio, it is one of the important formula, odds ratio, number of exposed cases. See here, exposed cases are 40 divided by number of unexposed cases, that is a 10. So 40 divided by 10 here in the numerator. In the denominator, number of exposed control divided by number of unexposed controls. See here, exposed control, unexposed control, that is 15 divided by 35. So you will get a odds ratio. It is very important in all the competitive exams also, and in the studies also. When you are presenting to a journals, writing an article, odds ratio is important. Keep in your mind. And these are some advantages and disadvantages of this case control study. It is efficient, save time and energy, used for very rare diseases and small sample sizes. You can be select. You can select only 10 people also. Okay, small, and you can generate a hypothesis, theory, just like a theory hypothesis. You can generate for future study also. You can generate, and disadvantages also there. Susceptible to a bias. What is bias? I will explain you. Wait. Okay, bias and recall, reporting, prone to methodology errors. Some errors may be possibility is there. Okay, and selection of appropriate comparison group may be difficult. Like that, some problems are there. This is called as a disadvantages that you can be note down and you have to write down in the exam. Okay. And interventional study, you come to the second part. Interventional study is also called as experimental study. Okay. So see here, see this table. Interventional study is divided into two types: quasi-experimental design, true experimental design. Quasi-experimental design, true experimental design. That we will see what it is actually. Okay, interventional studies, I have already told you, it is RCT, randomized, but here also it is also there. Don't get confusion. It is a randomized study and a quasi-experiment. Some studies it have a both names, two names. Okay, so don't get confusion. Interventional study is a two types here: quasi-experimental design, true experimental design. It is somewhat related to RCT, non-RCT. I will explain. Okay. First, we will see quasi-experimental design. See here, The name itself indicates quasi-experimental design is also called as non-randomized interventional study. I told no RCT, non-RCT. This comes under non-RCT. See, that's why name non-randomized interventional study. That's it. Don't get confusion. This is a synonym for non-RCT, not RTC. RTC means it is RTC bus stand. Okay. Don't go RTC bus stand to do any trial. Okay. RCT. Yeah. Okay. Only first point you can be noted. That's it. The remaining part you left it. Okay. Only first point is important. Quasi-experimental design. Yeah. Quasi-experimental design is again three types. Only one group will be there. Pre-test, post-test is design. Pre-test, before test, post-test, after tests. Okay, before test, after test. Just like a prepaid, post-paid. Prepaid means before talking itself, we are getting prepaid. We are paying to him. Postpaid means after talking, you are paying a bill. It is called a postpaid. So here also, before test, after test. Non-equivalent control group design. See here, name itself indicates non-equivalent control group design. Non-equivalent and case group is different. In previous slide, I have explained case are 50, controls are 50. Here, non-equivalent control. So I am taking only 10 cases and 50 cases I am taking control. 50 are control, 10 are case. Like that, both are not equal. Cases are not equal to control. Okay. And interrupted time series design. See here, name itself indicates. Okay. Interrupted time. So I am doing a research not regularly. Okay. I will take a Sunday break. I will take a Thursday break. Okay. Like that. Interrupted time. I am interrupting the time. Because it is for a human being, it is not possible to do continuous study. It is a type of study, interruption. You are giving some interruptions, either a 1 week, okay? Out of 12 months, 1 month you are giving an interruption. Like that, any day or any month, okay? Yeah, first one. I have told no, quasi-experimental design. One group, pre-test, post-test design. Here, only you write this example in the exam. Pre-test, post-test. Example, sample of violent adolescent women. So here in this experiment, they took ladies which are violent in nature. Okay. And they gave a treatment. What is a treatment? Anger management class. Anger management class. Okay. They took a class. How to manage the anger? Because they are very anger. That's why they are very violent, very violent. Okay. So that violent, to suppress them, violent, you are taking a treatment. You are giving a treatment. What is the treatment? How to cool, how to calm down, how to anger, how to manage your anger. And results, measures. See here, aggressive behavior in 1 year, pre and post. So pre-treatment, how she is behaving, how violently. After the class, after the treatment, how she is violently. Pre-test, post-test. Before the class, after the class. Okay, like that you are doing a test. Very simple to understand. All the study designs are very easy. If you're designing a study, okay, very nicely, you will get a pakka results. Either is there any changes in the aggressive behavior is decreased or increased, we don't know. Okay, that we will conclude with the result. And second, non-equivalent control group study. I have already told you. So this slides is already there. See here, pre-test, post-test. This intervention and activity is usual. Case number of cases is not equal to number of controls. That is the main point you have to keep in your mind. Non-equivalent control. The remaining, the schematic diagram not required. Okay, don't practice this. Already it was there in previous slide. Okay, you explain only number of cases not equal to number of controls. Interrupted time series design. Here also, time series analysis is simply a set of measurement of available tasks at various points of time. See here, Example, mood of students during the semester. So during the semester, only it is not possible to maintain one mood. Mood is go on changing, changing, changing. How the mood will be there? Okay, that was the example. See here, uh, table 1, table 2, or table 3, 4, 5, like that. And here I gave the X, interruption I gave sometime. I observed first 4 days, I gave a 5th day break, and 6th, 7th, 8th, 9th, I observed. That's it. Okay. Yeah, here you come to the most of the students, they do not know clearly what is accuracy and what is precision. Keep in your mind what is accuracy. They will think that accuracy and precision are same. Some people will say it is not equal. Here I will get a clear clarity regarding accuracy and precision. See this diagram. Here this is a target, and you have a totally 5 bullets. See here, one person has shooted 5 bullets here in the first one. So all are very closer, but it is not closer to the target. Keep in your mind, all the five shots are closer to each other. See here, observe the 5 shots here, closer to each other, but it is not closer to the target. So then you are calling as a high precision, but low accuracy. Low accuracy, high precision. Precision is high, very closer together, all 5 values. Okay, but accuracy is not there, only precision is there. See here, high accuracy, all 5 shots are there. All 5 shots not closing to each other, all 5 shots are close to target, but all 5 shots among them not closer. And you see here, both are high. Accuracy is there, precision is there, both are there. You keep in your mind. So whenever you are doing a study, your values, your results must be high precise as well as high accuracy. High accuracy, both must be there. This is very important. So here the winner is he. Here he is not a winner, he is also not a winner, he is a winner. Exactly on the red mark, all 5 shots are there. Precision is there as well as accuracy is there. Okay. RCT. This is also only heading you read, randomized control trial. Keep the name. Okay. No need not to be take a screenshot for this slide. Okay. Randomized control trial. See here, the name itself indicates random. Random. See here, you observe this thing. Here I took a treatment A, treatment B. 2 groups I have to. Treatment A, treatment B. And whenever I am these 2 groups I am giving, I have to randomly assign in the subject to a treatment. If suppose a person volunteer came, randomly I won't think much. Just when a person came volunteer, simply I will put as a either you go for A or go for B. Randomly. Okay, no logic, nothing will be there. That's why it is called as a randomly. Just you are taking it and putting into some A group or some B group, like that you are putting. That's it. Okay, there is no logic, nothing. See here, I have divided into two groups. Here you can see all the dots are there, blue color dots. All these blue colored dots, these people are called as a treatment A. I am giving a tablets A here, and I am giving a B tablets to these people. They do not have a blue color dot. So only to symbolize that. Okay, and here you can see some people with this A, they have developed a side effect, these red marks. See here, red color with blue dot. They got a side effects due to A. Here, they got a side effects due to B. They do not have a even. So like that you can remember. Red color stands for those persons feeling side effects. And yellow color, okay, they are good, no side effects. See here in the group A, only 2 persons are, they got side effects. Okay, here 1, 2, 3, 4, 5, 5 members are in red color. They have a side effects was for B, drug B, side effects are more. For drug A, side effects is less. We can conclude. So which is safe? A drug is safe because only 2 persons are there. Okay, 2 persons, only 2 persons got a side effects. It is a safer. Like that you can conclude your studies. And advantages or randomized advantages of randomized control RCT design. Comparative, we can compare. See in the last slide I have shown how to compare with yellow, blue dots like that. Minimize the bias. Minimize the, it is very important. Bias must not be there. Bias must not be there. If bias is not there, it is good study. Keep in mind. Most of the things students will thought that there must be bias. No bias. If no bias means it is a good study. And minimize the confounding factors and statistical reliability will be there. These are the advantages. Just you can be, all these headings are important, not the sentences. If you want, you can be write in the exam. Comparative, minimizing the bias, okay, and minimizing the confounding factor and statistical reliability. 4 headings are important. So now we will see what are the different types of RCT. Yeah, see here RCT, randomized control clinical trial, includes if you are doing any diagnostic studies, therapeutic studies, polyphyletic studies, okay, prophylactic means prevention. Keep in your mind, prophylactic means prevention is always better than that. So daily a person is consuming a B complex tablet to avoid the neuronal damage. Okay, that is called as a prophylactic use. Okay, those are called as a randomized controlled clinical trial, randomized controlled field trial. Field will be there. Field example also here I gave the example also. Only headings you keep in your mind. Preventive trial, prevention. Vaccine, best example to avoid the coronavirus. Vaccines are, it's a preventive trial. Risk trial to find out the side effects. Okay, first one see here, diagnostic, randomized. Second one, randomized control field, you are in the field. And third one, prevention trial like a vaccine. And fourth one, side effects, risk factor. Is there any risk of taking a tablets? Okay. Further classification of RCT. This is also a very important one. Here you will get all these 4 names you can keep in your mind. Open RCT, single blind, double blind, triple blind. A number of times this was appeared in the question papers. Okay, so you concentrate on this. It was already appeared in the old question paper. What is open? What is blind? What is double blind? What is triple blind? Okay, that I will explain you with the diagram. In the next slide, I will explain you. See here. Open trial. See here. See the cursor. First one. Open trial. Open trial means, see here, whenever you are doing an experiment, whenever you are doing a clinical trials, there must be, see here, medicine. See here, this is a medicine. These are all common. Medicine in any type of study, first medicine will be there. See here, staff is giving a medicine to the patient. So he is a patient here. He is giving a, staff is giving a medicine to the patient, and analyst is there. He is observing either what happening to the patient, and he is recording the result. So all these are the 5 steps: medicine, staff, patient, and analyst, either a doctor or Pharm.D doctor, okay, or MBBS doctor. And here he is drawing finally with the help of a computer. These are all very common. See, in open trial, Nobody is blind. Nobody is blind. All are opening their eyes. Why it is called as a opening? Means, see here, all the persons, all the three persons, they know what the medicine they are taking. What the medicine name, they know. What is the name of the medicine? Clearly they know, all the three persons. Whereas in single blind study, see here, blind, the patient is become a blind. Because why it is blind means he is not blind, actually he has a eyes. Okay, he blind means he do not know what is the name of the medicine. Okay, don't think that the blind patient, no, he has a vision. Okay, everything is good. Why it is called blind? He do not know the name of the medicine. You got my point? That's why. Okay, they are giving, it is a painkiller, you take, your pain will be reduced, like that they will give you. Okay, either it is, but exactly, either it is paracetamol, is it aceclofenac, is it a diclofenac, they will not tell. Only you take this medicine, it will be good for health. Okay, it is a painkiller, like that they will tell, but what the drug it is, they will not tell. And see here, double blind. In the double blind study, staff do not know what is the medicine name, and patient also do not know what is the medicine name. It is called a double blind. And triple blind, three persons they do not know. That's why it is a triple blind. Just company researchers, they are doing to do assay on these tablets. That's why it is called a triple blind study. Nobody knows what is the drug. It is only the company owner know. Okay. So now we will back to our slide. Open. In open RCT, everybody involved in the trial known which intervention is giving to each patient. Single blind, patient or evaluator, only one blind is there. Either patient or evaluator is blinded. Okay, only one person. Double blind, neither patient nor outcome evaluator knows. And triple blind, patient, physician, data analyst are blinded. They do not know what they are. Now I think it is a very clear cutly it was mentioned. Okay, I have explained with clear cut. Okay, and according to participations and exposure, I have already told parallel design and crossover design in my previous video. Go and see my previous video. I have already explained parallel design and crossover design. Okay, that two are important. Cluster design, fractional design is there, but it is not much important. Parallel design, this slide is more than enough. Okay, parallel, what is this parallelly? I have already told, go and see my previous video. Advantages and disadvantages of parallel design, you can be note down or take a screenshot and you can be write it. The what I have explained there in that video. Crossover design, this slide is more than enough. This diagram you have to draw in the exam. Okay, so that the evaluators, they will get impression that they have a cross. See here, cross studies. It is a parallel study. Okay. And cluster design, it is a cluster. Name itself indicate it is very complicated. Very complicated. Okay. See here, 10 hospitals are clustered together. Not, you are not doing in one hospital. 10 hospital. Cluster randomization. 5 clusters like that into 2. Okay, and control 5 hospital, intervention 5 hospital, and total study population is 1000, like that. N control 500, intervention 500, like that they will design. And mega trials, it is not much important. N of trials, N of trial means just 1 or 2, 3, 4, like that. Mega trials, mega, name itself indicate, just like mega star. Okay, mega trials, more than 10,000 subjects is called as a mega trials. And sequential trials, sequentially they do. That's why it is called a sequential trials. Yeah, so I will stop here. If you have any doubts, you go through, you repeat the video, rewind the video and go through, you practice. In the examination, you draw the schematic presentations, what I have shown. Okay, Later, you can show your talent under schematic presentation. First, you draw a schematic presentation very neatly. That is very important in the statistics and the research methodology subject. Okay. Draw a schematic presentation, and later below that, you show your talent in the JNTU or whatever it may be. You can show. Okay. You can perform, you can access it. Okay. And bye. Thank you very much.